greetings session. This is the final session of the conference. I just wanted to remark on how wonderful it has been for us to have such a great turnout for this online conference, and that I hope that everyone has had as good of a time at this online conference as I have. My name is Helen Tyne. I'm a research scientist at Agriculture and Agri-Food Canada. I'm the chair of the breeding session. I just wanted to say that there were very many great abstracts that were submitted for presentation at our session. We had to select, Ilan and I had to select just a very few abstracts for presentation, but they were excellent. Everyone's suggestions and abstracts. One sort of organizational thing that I wanted to mention to everyone is that our session will be followed immediately by the closing remarks for the online conference. The discussion group for the session, for the breeding session, will happen at 1825 Central European Time after the closing remarks. So don't go away after the closing remarks. Please join us in the discussion group that will follow the closing remarks in the link that was provided to you in the instructions and abstract book. Okay, so without further delay, I want to introduce the co-chair of our session, Ilan Caron. He is at the Institute of Plant Sciences of the Volcani Center in Israel. So Ilan.
Okay, thank you. Well, we will get started with the first opening lecture, Professor Dani Zamir. Dani is a professor of genetics at the Hebrew University of Jerusalem. Dani is a world leader of tomato genetics and constructed many genetic resources that are widely used by the tomato community. Dani combines very successfully innovative academic research and practical breeding of tomato, Isianthus, and roses. And for his outstanding work, he received earlier this year the prestigious Israel Prize for Agricultural Research and Environmental Science. So with that, I will turn the mic to Dani, please.
Thanks, Ilan. So now I have to share the screen.
Share the screen, yeah.
And this I do by?
The green button.
Okay, got that. Share. Share. So the PowerPoint is open, okay. Desktop.
Oh, we saw it flash up.
Okay.
Okay.
Got it.
Yeah, it's a pleasure to be here. And I'm going to tell you today about exotic genetic libraries, map resistance to the tomato brown rugose fruit virus, to the tobacco mosaic virus locus of tomato, the TM1. The work was done in close collaboration with Dr. Aviv Dombrovsky, who is coming from the ARO, Beta-Gan, and he's a specialist in virology and one of the first to identify this virus. And the work was done by Idan, who's a joint student, and the population that I will describe was developed by a student called Shai Turgeman. So I can't start a tomato genetics talk without talking about the grandfather of tomato research, Dr. Charlie Rick, who had the foresight to go and collect a lot of wild species accessions from South America long before people thought about the potential of wild tomato species for the crop improvement. He made more than 10 trips to South America and fell in love with this continent and brought a lot of accessions that are kept by the Tomato Genetics Resource Center in Davis and are distributed to whoever wants to grow them and play around with them. So this is the foundation of the exotic genetic libraries that I'll be talking about. Not only did he discover the wild species and made the collections, a lot of these collections, when you go down there now, you don't find them because there are cities and agricultural fields. But early in the 50s, I guess 60s, when he did the collection, he could find a lot of accessions. So there's more diversity in the Tomato Genetic Resource Center in Davis than in South America. He also developed the roadmap on how to utilize these genetic resources. And one of the first resources that he worked on was Solanum penellae that was collected in Atico in Peru from a desert region. And this was a favorite accession that was studied by the community, is still studied by the community. The F1 hybrid of the cultivated tomato with Solanum penellae is a huge monster. You can see maybe here in the middle, it's a huge plant compared to its parent. So it's obvious that there is a lot of vigor, a lot of hybrid vigor, heterosis, that is hidden in the combination of the two species. What was done in our lab by Yuval Eshed is to develop a set of introgression lines of Solanum penellae. And this is what we call the exotic genetic libraries. So we have on the top the Solanum penellae in green, because it's a green-fruited species. And then we have the variety M82. And through marker analysis and a lot of backcrossing, Yuval was able to cover the genome of penellae with 76 lines that cover the entire genome. The success of this population was derived from the fact that if you look at IL-11 and M82, they are nearly isogenic for all their chromosomes, except for the chromosome that was introgressed from the wild species. So it was possible to map genes that reside on the introgressed segment. However, since the genome is identical for the rest of the chromosomes, there were no additional noise that affects this trait, and no additional QTL or epistatic interaction that affected the trait. So it was possible to fine map it using the introgression lines and clean up the huge effect that epistasis can have. But being biologists, of course, we would like to move ahead and look also at epistasis. So on the left, you see the drawing, it's a drawing of Waddington, who show the pegs are at the bottom are the gene, and mountain, the topography is the phenotype. And there are rare cases where a single gene pulls on the topography and shapes it. In most cases, it's a few genes that interact together to uh, create a vector that pulls on uh, this topography and shapes it. So it's multiple genes that interact with each other to form the phenotype. And we would like to understand now these epistatic effects. For this, it was necessary to design a different population structure to handle epistasis. And there are two main factors that limit our ability to deal with epistasis, to genetically dissect epistasis. One is that usually the mapping populations are rather small, 100 or 200 individuals. So if you look at such a population in F2 or in other generations, it's rare that you find two individuals or multiple individuals that have two genomic segments together. And if you want to look at the interaction of these two genomic segments, you need a lot of at least 10 or 15 individuals that are independent and carry this segment. So one problem is that population is too small. The other one is the multiple testing penalty, because you have each locus, you look at its interaction with the rest of the loci that measured throughout the genome. So there are a lot of comparisons being done there. And for this, one has to compensate for the Bonferroni or other ways to compensate for multiple testing penalty. So when you compensate for that, there's no epistasis. Of course, epistasis is always there, but we have to find a way to dissect it. So we found a new population, a new accession of Pinelli. We call it the lost accession. You can see here it, in the field, first time we saw it in 2007, where we grew the core collection for the EU Soil project. And one, it was all cultivated tomato, but one of the envelopes apparently contains seed of Solanum Pinelli, and it was beautiful Solanum Pinelli. It could go well in the field, something LA716 did not do. Another thing, it set self-seed very easily and had developed root system, etc., that made it an attractive candidate to build a new population. So what we did is we took Solanum Pinelli at the top of the triangle, the green-fruited species. We crossed it to Lea, which is a determinate tomato variety, and top, which is indeterminate tomato varieties. We made the F1 hybrid, then we made backcross 1 and backcross 2 to the cultivated tomato. We had approximately 2,000 individuals with the Lea and 2,000 individuals with the top. And then we started selfing it down to self-6. Well, we lost quite a bit of lines due to sterility, but still in the lab background, we had 1,500 backcross 2 self-6 lines, which is a considerable number of individuals. After that, in the framework of the G2P Sol, we were able to generate a map for the population. you can see the 12 chromosomes. If you look at chromosome 1 on the left top, you can see the characteristic region. On the left is the euchromatic region that is rich in recombinations. Then there is the centromeric region, a few recombinations for the physical distance, and then the other arm of the euchromatin where there is a high recombination. What we found that the population is highly recombinogenic in the euchromatic regions where we have 18, we have on the average 18 kb defined by the different lines in the population. So we saw that the high frequency of recombination that can resolve segments of 18 kb seemed a bit fishy to us at the beginning. So we used the PCRs and we looked at two neighboring genes along the genome, and indeed we found a lot of recombination between neighboring genes. This is in the euchromatic region. If we look at chromosome 2 has one has a centromeric region here and euchromatic region. And you could see that the transition between the heterochromatic to the euchromatic region is around 35 million base pairs on the chromosome 2, etc. You could see the entire genome on the map. Okay, so what the next thing to do was to look for epistasis, and the way we did that, we planted in the field the Becros 2 cell 5, 1,500 lines, and Becros 2 cell 5 was crossed to Lea, so we had the bill hybrids. The bills were homozygous for the introgression coming from the wild species, whereas the bills crossed to Lea gives the bill hybrid that are heterozygote for the introgress segment, usually much better performing than the bills themselves. And here you can see one of the screening trials. These plants were irrigated about 20% of the amount that usually processing tomato require. And you see the cracks in the soil. And every once in a while, a tomato would fall down in the crack. You probably hit somebody on the head in China or the other part of the planet, because these cracks are very deep in the soil. So we phenotype the population, looking mainly, of course, at yield. And we did multiple experiments, and you can see that there are genomic regions, like in chromosome 4, there is a genomic region that increases yield in drought in 3 different populations that involve these causes. So one can map using this resource individual QTL, but what happens to epistatic QTL? Well, it's possible to run for epistasis, analyze epistasis by computer and looking at regions where there is a bright segment indicating high load for epistasis of segments in chromosome 7 and segments on chromosome 1. And when we planted this material in the field in 2019, so you could see on the left are the lines that are homozygote for the cultivated tomato allele at both loci, chromosome 1 and chromosome 7. So they have no introgressions here. The average yield was around 3.5 kilograms per plant in the drought condition, whereas usually it's 8 or 9. Plants that were heterozygote for the first locus had very similar yield to the population itself, and plants that were heterozygote only for the chromosome 7 locus had also very similar yield. But when we have a hybrid that carries both introgression, there is a significant increase in productivity, around 5 kilograms per plant. And you can see that we can call it epistasis because this one segment does not have an effect, the other segment does not have an effect. And when you put both these segments together, you have a large effect. I'm hiding the, but here you can see that we were able to reproduce this effect in 2020. So the lines without the introgressions were very similar to each other, whereas lines that were heterozygous for the introgression had a significant increase in productivity. So this element, I see that my time is running out. So anyway, this element of epistasis we are continuing with. While we were doing the crosses in the greenhouse, preparing the seed, it was a heavy season for the tomato brown rugose fruit virus, which is a tobamovirus member of a family like the TMV. This virus breaks the resistance of TM2A. And here you can see the symptoms. The leaves are wiry-like. You can see these shadows, these spots on the fruit. Sometimes the fruits turn brown. I don't know if it's the virus. It's one of the characteristics. The brown is one of the characteristics of the virus. So it's very ugly. The disease is spreading very quickly in a greenhouse that is infected. We saw in our bill population, first of all, Penellae was not affected by the disease. And the other thing, there were individuals in the population that had no symptoms and had no virus. So that was kind of exciting. And since we had the genotype of the plants from previous analysis, we could score the phenotype and we could identify QTL that improve the level of resistance of the population. One is on chromosome 2, another one on chromosome 9 with a smaller effect, and on chromosome 11, there is one that has, it's not my head, here is the one that has a negative effect, makes the plants more susceptible. So this is the concept of the genetic library. You make a population with a wild species, progression line, Becker's inbred line, that is fixed. You run the markers on it, and you keep it on the shelf. And we have in the lab 10 such populations with 10 different accessions, but we always make additional populations because there are many that Charlie collected, so it's a challenge. Anyway, the fact is that once you have a new disease, and there's endless diseases, then you go and you pull out the parents of the population, you inoculate them, you find the one that has the resistance, and immediately you have the mapping. So this is the mapping of the QTLs for the resistance, and the QTL were affecting the resistance in both background, in the layer and in the top. You see that high number is susceptibility. So you can see the ones that are the ones that carry the cultivated tomato allele is much more susceptible from the three that carry the Penelli allele. And so is the story for the top background. So when we go back, you see some of the QTL that we detect, this one on chromosome 9 or other ones that were detected, were specific for specific populations. Whereas the chromosome 2 was effective, all the time, and using the high number of recombinations that we have in this region of chromosome 2, we could map it to about half gigabase. And this is the region where the resistant resides. And this region also included the TM1 gene that was cloned and identified previously. So the TM1 originates from Solanum habucaitis, the historical TM1. In 1957, the introgression was made by Holmes. And later on, a group in Japan did a wonderful study that shows that the gene product of TM1 inhibit viral RNA replication, and it maps to around 34 million base pairs on the physical map of the tomato.
Danny, you have two minutes.
2 minutes is good. And so up till now, we talked about the Penelli allele. So of course, we said, okay, let's test the hirsutum allele. And in the Tomato Genetics Resource Center, there are multiple lines that have the TM1 gene. And also they show that lines that are free, that carry the wild species allele, are much more resistant to lines that carry the cultivated tomato allele. So we have now the TM1 of hirsutum or of habucaitis origin, and TM1 of the Penelli origin. And the disease is appears, so it's not coronavirus, but in the literature of the tomato is a severe disease, makes very ugly fruits. But I believe that in a year or two, the progress that is made by seed companies in developing resistant varieties will lead to solving this problem of the rugose, and there are a number of patents on this for different companies. So to close, I say that we want to improve plant breeding, and that's the objective, whoever studies plant breeding research. We want to do it, and I follow Dr. Rick in saying that exotic genetic libraries is the way to go. There is a paper I published in 2001. There were GMs, and there is the CRISPR, and a lot of other miracles that every once in a while scientists get excited about, and indeed it's exciting. But for improving plant breeding, I believe that exotic genetic libraries is the solution. Thank you very much.
Okay, thank you, Danny. I don't see on the chat any question, so please just
But there is one question here, Elon.
Oh, okay, sorry. Are there differences in the level of resistance between the TM1 of Abursitis versus Spinelli?
Yeah, of course, it's a major question that we are testing this season. I mean, but this, all the experiments I described are experiments uh, that were conducted up to this year. So it's all, but there are, of course, a lot of questions that are open. One of them is comparing the alleles of Abrikitis and Pinelli. Another important question is symptoms. The symptoms can occur in the fruit, can occur in the leaves, can occur in other parts of the plant. And the question is, will the resistance provide uh, protection for all organs of the plant? Still is a question.
Okay, maybe just one more question. Are resistance where the plants really are resistant or tolerant? Are the plants are resistant or tolerant? That's the question.
Well, I think they're resistant. Usually, they never develop symptoms. Sometimes you can detect using antibody. You can detect the virus or parts of the virus in the plant, and sometimes not. But it's not immune. It's resistant.
Okay. Okay, I think we have to go on because we're running already late. So thank you very much, Danny. And there will be a discussion after the breeding session. So there will be some more chances to you know, ask and answer questions. Thank you, Danny. Okay, so we will move to the next speaker, which is Sean Fenstermaker from Ohio State University. He will talk about using Solanum galapagense to achieve tolerance to water deficit through introgression breeding and grafting. Okay.
Okay. Hello, everyone, and thank you for joining me today. I'm a graduate student working with David Francis in the tomato lab at the Ohio State University. And today I'm going to present some work I've been doing with an accession of Solanum galapagense. Okay, so I want to introduce you to the wild accession I'm working with. This is Solanum galapagense accession LA1141. It's a wild tomato from the red-fruited clade. It was actually collected from the outcroppings of a volcanic crater on the Galapagos Islands, where there isn't a lot of water available. This accession is a source of unique pigmentation, high beta carotene, trichomes and secondary metabolites that deter insects, and also the focus of today will be water deficit tolerance. The objectives of this project were to determine the genetic basis of water deficit tolerance in accession LA1141. And to do this, we conducted QTL studies and also used genomic selection. We use this data to make selections, and then finally we wanted to test whether we can deliver the water deficit tolerance trait through introgression breeding or grafting. Here we are looking at accession LA1141 on the left, and the elite Ohio processing tomato, Ohio 8245 on the right. And we use these to develop an inbred backcross population. There were 158 individuals in the population. And as you can see, our parents are separated pretty well for the wilt trait and also the canopy temperature trait. And I'd like to just remind you that canopy temperature is really a proxy for stomatal opening and closing. And that's controlled by the root sensing water. And this was really an ideal population for mapping because of the parental separation. Our experimental design allowed us to correct for spatial variation across the greenhouse. So some of the areas of the greenhouse are hotter than others, and that's caused by a temperature gradient between the cooling pad and the fans. So we have replicated controls in rows and columns, and this really allows us to correct for those spatial differences. And multiple greenhouse experiments were conducted to evaluate the response of the inbred backcross material. The traits were measured, the traits that we measured were canopy temperature and wilt, and canopy temperature was measured both with a handheld infrared thermometer that you see here on the left, and also using thermal image data that we collected using this iOS attachment from the company FLIR. And we rated wilt on a scale of 0 to 5. A rating of 0 is a dead plant, and a rating of 5 is a completely turgid plant. And in this image here, I'm showing an example of a plant that was rated as a 3. And these pictures show the steps of the process for the thermal image analysis. And then first we extract temperature data from the picture, and then we use a histogram-based thresholding method to estimate canopy temperature. And what I want to show you with this slide is that we see a higher proportion of genotypic variance when we use the handheld thermometer. And this suggests that the thermometer may actually be a better method. The thermal image thresholding methods are still being improved, and I look forward to being able to share data about these comparisons in the future. We used an optimized 384 amplicon-based SNP array to genotype the parents and the progeny in the inbred backcross population. 152 of those markers were polymorphic, and they were evenly distributed throughout the genome, and the genotyping was done by Agriplex Genomics. Here, I want to show you the genetic map, and you can see that the markers are pretty well distributed across the genome. We use the R package, RQTL, to create the genetic map for the population. And the average centimorgan spacing is 6.1, which approaches the ideal 5 centimorgan interval for being able to do mapping. And what I want you to take away from this slide is that as many as 7 putative QTLs were identified, and only 1 QTL was reproducible and replicated experiments for both canopy temperature and wilt traits. The effects of the allele substitutions were small. They lowered plant canopy temperature between 0.2 and 0.5 degrees centigrade, and for the wilt traits, they lowered wilt by 8 to 12%. We did detect a reproducible QTL on chromosome 5, which explained 5% of the phenotypic variance and decreased wilt by 8%. We use phenotypic data and genomic estimated breeding values to select progeny from the IBC population. The selection intensity for canopy temperature was 1 standard deviation above the mean, and the selection intensity for the wilt traits were 2 standard deviations above the mean. 6 selections were advanced for germplasm development. Our selections demonstrated tolerance in additional water deficit experiments. When we compare estimated breeding values to our phenotypic data, our percent co-selection ranges between 26 and 40% for canopy temperature traits. And for the wilt traits, it's between 32 and 33%. And for the remainder of the talk, I'm going to focus on SG18197. This is the line that has the putative QTL on chromosome 5 that's associated with the water deficit tolerance. So we thought that integration in this IBC material would be a challenge because the allele substitutions had such a small effect and they explained a low proportion of phenotypic variance. And it would be risky as a breeder to integress many small effect QTL from wild germplasm, while at the same time maintaining important traits such as fruit size. And because our lab has a rootstock breeding program, we decided to explore an alternative method of trait delivery by using this material as rootstock instead. So what I'm showing is we screened 71 heirloom and landrace varieties for performance under water deficit. And we chose Juan Flamme because it performed pretty poorly in the water deficit experiment. And also it's popular among high tunnel growers locally. And we thought it had potential to be improved with grafting. So in replicated greenhouse experiments, we do see a significant effect on scion wilting when Juan Flamme is grafted to both the LA1141 parent and also our IBC line SG18197 compared to both self-grafted and ungrafted controls. And the water deficit tolerance derived from LA1141 does appear to be graft transmissible. So the takeaways from this work are that the genetics of water deficit intolerance derived from LA1141 is genetically complex, and integration would be difficult because of the small effects of allele substitutions. Heritability of the canopy temperature trait is higher when we measure plants using the handheld infrared thermometer. And phenotyping and genomic selection identified progeny that has tolerance to water deficit. Our selection, SG18197, possesses the putative water deficit tolerance QTL on chromosome 5, which is derived from LA1141. And when used as a rootstock, LA1141 and SG18197 significantly delays scion wilting compared to self-grafted and ungrafted controls. And ultimately, grafting may be more effective than integration to deliver water deficit tolerance from LA1141, at least in these experiments. And I would like to thank my lab members and my advisor and our collaborators that assisted with the development of the inbred backcross population, and also just acknowledge my funding source, which SCRI grant from the USDA. And thank you for your attention. And I just put my email up there if anybody wanted to reach out.
Okay, thank you very much. Are there any questions? I don't see questions on the chat. Okay. The link for the discussion after the meeting, after the session, is pasted now. So everyone is interested, we can access it.
I see.
Okay, well, we'll move to the next speaker, which is Yuri Tikhonov from Wageningen University. He will talk about the genetic and functional analysis of flavor in commercial tomato and novel floral 4-gene underlies QTL for floral aroma volatiles in tomato fruit.
Well, thank you, Ilan, and I hope everybody can hear me and see my front slide.
Yes, we see.
All right, so, well, first of all, on behalf of our group, I would like to thank the organizers of this meeting to give us this opportunity to present our work in front of the Solanaceae community, and for the opportunity to finally ask all of you whether you ever had a tomato fruit that actually tastes like rose flower. Well, probably not, but if you ever have, please let us know in the comments and we will contact you later. So, and then, well, what about these substances then? So have you ever had a tomato that tastes like any of these? So probably also not, or maybe some of them, but I can assure you, if you take all these components in the correct proportions, put them in an old suitcase, close it and knock it down the stairs of a high building, at the bottom, you may get something that resembles tomato flavor. Well, and this is exactly the experiment that I'm not going to talk to you about today, maybe next time. So alternatively, you can play with different chemicals that are responsible for all the flavors that and odors that I just showed you. And in fact, in practice, it's done by companies, especially by food companies, which create different food additives. But as breeders, of course, we would like to be able to control flavor in the fresh tomato fruit. And to do so, well, there is a very simplified scheme. So you can basically deconstruct your tomato flavor for different components. Like you see here, you can screen your materials for these components. Then you can find, so by chemical profiling, you can find the chemicals which are responsible for these individual compounds. By merging this data with genetic data, you can find the QTLs which are responsible for these chemicals and flavor trace. And at the end, you can come up with genetic markers that breeders can use in the practice, or you can make it a little bit more complicated to add some functional resolution into your analysis and go for enzymes and genes which are responsible for these chemicals, and produce more, let's say, precise markers. So, and of course, what you need for that, you need diversity. So, and that's what we have started with a long time ago. We started with the 94 cultivated greenhouse cultivars that we acquired from different breeding companies. So the fruits of these cultivars were subjected for different phenotyping and genotyping exercises, including sensory panel, different metabolomic platforms, and a SNP array, which contain about 5,000 different SNP markers. Then all this data together were subjected for diversity and QTL analysis. And what we did, we basically looked at the genetic and metabolic diversity. We already at that point, we produced some associations between markers and traits. But to validate them, of course, we selected four different parental lines that basically represented the genetic and phenotypic diversity of this set. We crossed them. So two of them were cherry tomato, and two of them were normal-sized round tomatoes. So we made several crosses and studied these crosses in different generations by basically subjecting them back to all these screening platforms that I showed you. At the end, we came up with a map, with a QTL map, and you can see it here. A map for different QTLs for chemical and quality traits. And of course, you can say, well, there we go, another map. And you are right, of course, because, well, in principle, there were quite some studies already performed on this subject, and there are plenty of maps. And I can tell you that, well, in a way, our map also resembles all these old maps, although there are some new things there. But the breeders were, of course, interested in how well they can use this QTL information in their practical breeding. So we performed a kind of valorization exercise in which we acquired about 1,200 different tomato genotypes that represent the diversity of breeding companies' germplasm. And we genotyped them for 18 different QTL regions with approximately 10 markers per region. And we calculated the frequency of with which our parental haplotypes appeared in this breeder's germplasm. And what you can see here on this plot is that actually many regions are quite well represented in the breeder's germplasm, and therefore they have a very good potential to be used in practical breeding. One of the let's say strongest QTLs that we observed in this QTL map was a QTL on a chromosome 4, a QTL for 3 different phenolic volatiles. And what's common about these phenolic volatiles is that they all have some sort of floral, fruity, rosy type of aromas. And here you can also see that they also have very similar chemical structures. And this similarity in chemical structures basically comes out of the pathway that they are produced from. So they are all produced by catabolism of phenylalanine. Well, this QTL, of course, is not new. It was reported in the past in several QTL studies dedicated to tomato flavor. You can see the studies here. And collectively, the studies, they allocate this QTL around 52, 56 MB on chromosome 4, and this is quite similar to what we found in our study. And the gene which is underlying this QTL have been never reported before by these studies. Another example where we know the gene which underlies the production of these floral volatiles is, of course, a famous malodorous locus, which was found in the interspecific crossing population between a cultivated tomato and Solanium2020, which Danny was just talking about. So, and it's called malodorous because of very high concentrations of phenylacetaldehyde. And well, despite the original floral characteristics of this compound, at these concentrations, it gives very unpleasant flavor. And later on, the underlying gene of this QTL was identified by the group from Florida, and it appeared to be an aromatic amino acid decarboxylase. So one way to look at the, about the concentrations, as I said, in that study, in study of the malodorous locus, the concentration was about 10-fold higher than the concentration in the cultivated tomato. And in our study, well, we didn't know whether our concentrations had anything to do with the actual flavor of this fruit, of the tomato fruits. And one way to look at that is, of course, to look at the associations between sensory panel and chemical data. And we performed it by doing a multivariate regression analysis with a random forest regression. And what we found is that, first of all, the smoky volatiles, eugenol, methylsalicylate, and glycol, they had a very high impact on tomato flavor. And the second group was the floral volatiles, which you can see here, and they were positively associated with sweet aftertaste, sweet taste, and sweet aroma. But this is just a statistical correlation. So what about real association? Is there something known whether this compound really contribute to tomato flavor? And yes, it's known from literature. So, for example, this study by a group of Elizabeth Baldwin showed that phenylethanol enhances tropical and fruity flavors, especially in the combination with sugars and acids in tomato fruit. We performed our own exercise to get more evidence for this. And so what we did, we developed a couple of near isogenic lines that were contrasting in that region on chromosome 4, and also, as you can see, they are quite contrasting in the accumulation of the phenolic floral volatiles. And the sensory panel observed that these meals were also different in so-called rose hip aroma, fruity, and overall aroma presence. Although you can also see that if you look at the overall scores of this, so this is not, these flavors are not something fundamental for tomato flavor, which basically tells you that, well, you probably never will find a tomato that really smells like rose flower. So we decided to go to the underlying genes of this QTL, and the original size of the region that we estimated was about 600 kilobase pairs, which contained about 50 different genes. So, and that was still a bit too many for us. Therefore, we decided to go for fine mapping. I'm not going to the details of this, but it's basically done by searching for recombinants and screening them with metabolomics tools and S&P platforms. And this allowed us to come down to about 110 kilobase per region, which contained 11 genes. And well, if you look at the annotations of these genes, it looks like, well, almost none of them have anything to do with any metabolic function except one, which is overexpressed in some stages of the, let's say, a high floral aroma parent. And it's also annotated as 3-methyl-2-oxybutanoate dehydrogenase containing E1 component. And later on, we also performed some qPCR analysis of this gene to get, let's say, a better data, because the data which I showed you before here, this is microarrays. And with the qPCR, what we saw is that indeed in the high floral parent, the expression of this gene remains more or less constant during all the stages of the fruit ripening. And in the low floral parent, it goes down, and it explains the increase in the phenylethanol content of this high floral parent quite well.
2 minutes.
Yes, as I said, this gene contained the E1 subunit, and that's quite important because this gene basically can perform this very first step of phenylalanine catabolism, or in general terms, amino acid catabolism. But if you look at the annotation, it's supposed to do that with a kind of wrong substrate. So it's supposed to act on branched chain amino acids. Anyway, that was the best candidate that we had, and we started with the functional analysis of this gene by doing some reverse genetics, and we started with virus-induced gene silencing of fluoro4 in tomato fruit. And what you can see here, first of all, the reduction in expression of this gene, and also the reduction in the production of both phenolic volatiles, which are responsible for floral aromas, and also reduction in branched chain amino acid volatiles. So that's the assumed function, real function of the gene. So apparently from these exercises that could do both. So we also produced some CRISPR-Cas mutants, and they basically showed the same result, the reduction in floral volatile production and also in some branched chain amino acid volatiles. Then the remaining questions that we have, so whether we could actually annotate this gene as an actual decarboxylase, and we did some E. coli enzyme assays, although they failed, so we couldn't show any activity. And we assume this might be because this floral 4 is a part of enzyme complex, and by doing that in E. coli, we fail because not all the components are there. And also now more broad questions, so how much we can rely on the annotations of this gene when selecting candidate genes for our traits. At the end, I would like to acknowledge, of course, our group, and we recently published a paper in a Plant Journal, and here you can see the most important people for this. Rana, who was a PhD student and did some fine mapping analysis, Finn and Jos that are the essence of this project, Joao and Richard, who had some genetic analysis and bioinformatics support, Iris, who made transgenic plants together with Jos. Fatima was involved in the enzyme assays. Chris, that provided the random forest regression script. Mariska, Caroline, and Wouter did a lot of sensory analysis. Shaq and Fred were involved in the genotype and genetic analysis, and of course, Richard Visser and Arnold Bovie for their never-fading leadership. And this project was supported originally by Netherlands Center of Biosystems and Genomics, and then later on by the Dutch organization, which is called Top Sectors, that supports innovations and research. And of course, thanks a lot for all the companies involved for their funding, for their support with the materials and also some experiments, and never fading hope in what we have been doing. And thanks to all of you for attending this talk.
Okay, okay, Yuri, thank you very much. I don't see questions here, so we.
Oh, I think there were some questions in the Q&A box.
No, I don't see any.
I think Jose Padua. It was Yuri, great talk, thanks. The sensory panel for flavor is done by humans, or do you have any metabolomics, chemometrics approach to evaluate it?
Okay, sorry.
No, that was done by a real sensory panel consisted of 8 trained panelists.
Okay, thank you. We have to go on and continue with eggplant now. And the speaker will be Jaime Proens from Universidad Politécnica de Valencia in Spain towards the development of the first eggplant magic population and QTL analysis of selected morphological traits.
Thank you, Ilan. Can you hear me?
Yes.
So thanks. Well, I will talk to you about the development of magic population in eggplant, in which it's the first magic population of this crop. This is a work that has been done by our group in Valencia, together with some colleagues from a seed company and also from a postdoc from our group who is in Japan at present. Well, in eggplant, there are very few experimental populations compared to other crops like tomato. And up to now, there are a couple of recombinant inbred line populations between eggplant accessions. One of them was obtained by French groups in INRA, and another one by an Italian group at CREA. Then there is another population, which is an introgression line set obtained with Solanum incanum, which is one of the wild relatives of eggplant, which separated 1.6 million years ago. And it consists at present of 53 introgression lines. It is not yet complete. There are some gaps in chromosome 6, in chromosome 11, but we hope that this set that we started some years ago, we will be able to finish it soon. Then there are new sets of introgression lines that are being obtained by our group following the the idea and its inspiration by Danny Samir about creating exotic introgression libraries. One is with the wild ancestor of eggplant, which separated around 6,000 years ago, in which we are in the BC4, almost is finished. We have to sell the plants. Another one is with Solanum dasyphilum, which separated 3.4 million years ago. It's from the secondary gene pool. We are also in the BC4. And another is very far away from the eggplant gene pool. It's with Solanum elagnifolium, which separated around 6.7 million years ago. We are a little bit more delayed with this one, but we hope that in a near future we will have this introgression line materials. And in fact, these introgression lines, we have already been observing some new phenotypes. For example, here you have the phenotype of the eggplant parental with long and purple fruits. They have anthocyanins, but no chlorophyll. They have no prickles. And in some of the advanced back crosses, we observe new phenotypes, like one which lack anthocyanins. So we know the genomic region where a major anthocyanin gene is present. The same with chlorophyll. We have some materials that have anthocyanins plus chlorophyll coming from the wild ancestor. But also, amazingly, some of the advanced back crosses have round fruit, like the wild ancestor. So there is a lot of genes of interest that we will be able to map and to find even the gene. Also for prickles, the cultivated parent has no prickles, and we have several lines that have the prickles typical of the wild ancestor. However, despite the interest of the new populations, we thought that it would also be very interesting to have a multi-parent population. So we decided to create a magic population, which is the first one in eggplant. And this is an 8-weight magic population in which we selected one wild parent, which is Solanum incanum. It comes from Israel. It's a wild plant very tolerant to drought. And then 7 genetically and phenotypically different eggplants. So we used this type of approach, looking for genetic variation, geographic variation, and also phenotypic variation. So one of the first things that we did, it was in collaboration with the group of Aureliano Bombarelli, was the resequencing of the parents. It was an Illumina sequencing at a depth of around 20x. So we found more than 10 million variants, of which 9 million were from the wild relative, and around 1 million for each of the cultivated parents. Then in the phylogenetic tree, of course, the wild species was completely differentiated from the others. But when we looked at the PCA of the cultivated parents, some of the parents were closer than we thought. So some of them were quite similar, despite being phenotypically different. So the scheme that we used for the magic population is a 8-way funnel. So we started with the 8 parents. We constructed the simple hybrids. With the simple hybrids, we obtained the double hybrids, and we used 298 double hybrids, 149 of each of the double hybrids, and we crossed them using a chain pollination scheme. So in the end, we finished with 265 crosses, and we used 2 plants per cross. So from these 530 initial plants in the S0 generation, we started a single seed descent program in which we have been using 2 plants per family to increase the probability of obtaining progeny of at least 1. We just keep 1, but we put 2 plants to ensure that we have at least progeny of 1 of them. our target is to obtain 384 S5 lines. So we are already in the S3 population. We have 421 individuals that were genotyped using the 5K probes SPET eggplant platform, which was developed with the Italian groups of the University of Turin, CREA, and ENEA in the framework of the G2PSOL project. And we obtained over 22,000 SNPs, and after filtering them, over 6,000 SNPs that were used for this population. The number of SNPs per chromosome varies between a little bit over 200 for chromosome 7 to around 1,000 for chromosome 1. Then, as we had the S3 population, it was just single plants, but we decided to score them for 5 very simple traits in a binary yes-no fashion. So we looked at presence of prickles or not, the plants that had anthocyanins, if they had the purple color under calyx or not, if the fruit had a flattened fruit shape or not, if the color of the flower was white or had some anthocyanins, and if in the peel of the fruits we had anthocyanins or not. So in the genotyping results, we got that the heterozygosity of the population was 6.33%. So it's quite homozygous, but after 2 more generations of selfing, we expect to be highly homozygous, 1.58% in the S5. We performed a PCA analysis in which we observed quite a broad distribution of the individuals in the graph, with relatively low values for the first and second component, indicating that there is low genetic structure. And well, in the MAGIC population, we observed a wide diversity with new phenotypes, both for the fruits, for the flowers, and for many other traits. So by combining the genotypic and phenotypic data, we made a GWAS analysis, and we found SNPs associated to the traits evaluated. Some of them, which are marked in green, here, for example, for prickles in the chromosome seeds, colocalize with previously detected QTLs in different populations, as F2, REALs, association panels, made by the groups of the University of Turin, the CREA, and also by Turkish and French groups. So we detected already known QTLs for prickles and also for purple undercalyx, but some new one QTLs for prickles, for fasciated fruit, and also for white flower, as well as confirming another one for anthocyanins in the fruit. Well, our next steps are finalizing the MAGIC population. At present, we have 289 lines and 187 lines, which is roughly 50% of our target. So we hope that in 1 year or a little bit more, we will have the final MAGIC population. And also an important thing is that we can take advantage of the advanced highly homozygous S3 lines that segregate for specific traits. As I mentioned before, for each progeny, we use 2 plants. And sometimes we observe segregation, like here, in which 2 full SIP plants segregate for presence of anthocyanins and lack of anthocyanins. So this is very useful to develop near introgression lines. Another next step is the high throughput genotyping and phenotyping of the final MAGIC population, and of course, selecting some improved recombinant material in this population. In conclusion, the MAGIC population has a high diversity with new phenotypes and lack of evident genetic structure, which will facilitate detection of QTLs and selection of recombinant materials. The level of heterozygosity is already low in the S3, indicating that in the S5 we will have a highly homozygous population. We have detected several SNPs for simple binary traits, and some of them are new and others are associated to former QTLs, but it has been possible to narrow down the QTL region. Also, the introduction of a wild parent in the magic population may reveal the genetic basis of domestication and stress tolerance traits. And together with other experimental populations, this new magic population will be a powerful resource for eggplant genomics and breeding. Also, if you are interested, we published very recently a review on the use of magic populations in plant breeding that you may consult if you have an interest. And finally, I want to thank the people of my group who participated in developing this magic population, and also the Spanish government and the European Union through the G2PSOL project for funding this work. Thank you.
Okay, Jaime, thank you very much for the very nice presentation, nice eggplants. I see there's a question, why does the IL line without anthocyanin but with chlorophyll still look blackish? And is it the case that the black eggplant comes from combined presence of anthocyanin and chlorophyll?
Yes. The black color of the eggplant is the combination of anthocyanins plus chlorophyll. So when you have both pigments, you have black or dark purple fruits. If you have just anthocyanins, normally is purple, and if you just have chlorophyll, it's green. And if you have none of them, it's white.
Okay. And a second question, what is the preliminary QTL results for chlorophyll in the fruit?
Well, I have not presented here, but we also found a very strong QTL for chlorophyll content in the peel, and we have narrowed down quite a lot the genomic region, and we have several candidate genes for the chlorophyll content. So with this population, we have also been able to spot the QTL for chlorophyll.
Okay, and just last very short question, and should be a short answer. Are eggplants autogamous?
Well, they are self-compatible. They are usually autogamous, as we have seen in the genotyping of materials from gene banks. But if you have bumblebees or other pollinators in the open field, there is intercrossing between them. So it's mostly autogamous.
Okay, well, thank you, Jaime, and we have to move on to the potato section.
Thank you. So I have to stop sharing.
Yeah.
Okay.
All right, thank you, everyone. I think we're going to do something a little bit different for the next two talks. They are going to be presented by Ernst-Jan Eggers from University of Wageningen, and then followed by Natalie Kaiser from Michigan State University. We're going to ask you to save your questions for both of these talks. They're both related to diploid inbreeding in potato and the SLI gene. We'll ask you to save your questions for Ernst-Jan so that they occur together after Natalie's talk. So if you have a question in the Q&A, please indicate if you would like to address this to Ernst-Jan, Natalie, or both. So without any further ado, please, Ernst-Jan.
Well, hello everyone. My name is Ernst-Jan Eggers, and I work for Solinta, and I'm doing my graduate research in Wageningen University. So at Solinta, we do diploid hybrid potato breeding, and we propagate our materials through potato seed. And our main aim is to develop potato inbred lines, which allow easier genetics and can produce uniform F1 hybrids. Not so long ago, we released Solintas to the scientific community and sequenced it quite deeply. But of course, we're not the only group doing this. So recently, Hosaka and Sanetomo published about inbred line they created. In 2014 already, Shelly Jansky and Kipa Padukal released M6, and also an inbred line.
And in China, there is also a lot of inbred line development. And after my presentation, Natalie will tell more about her work in her lab. So to create inbred lines, you need, of course, self-compatibility. But most diploid potato accessions actually show self-incompatibility, which means that self-pollen is not able to grow through the pistil and is not able to self-fertilize. So this is due to the S locus, which encodes style-expressed sRNAs and pollen-expressed F-box proteins. And the idea is that if these sRNases are not recognized by the F-box proteins, they exert their cytotoxic effect on pollen, calling pollen tube growth arrest. So there is a bit of an extension to this model. So we know that the S locus contains multiple S locus F-box proteins. And the idea is that together, all these F-box proteins can recognize all sRNases except the one that is present on its own allele. Well, luckily, there have been reports about self-compatibility in potato for already a long time. So Alzer and Hampson published in 1976 their finding of a self-compatible line. More recently, well, in the 90s, Hosaka and Hahnemann published about the SLI gene, the S-locus inhibitor gene. And they mapped the SLI gene to chromosome 12. Then really recently, Plot et al. Published that they find this gene quite widespread in potato, and they confirmed its position on chromosome 12. So at Solinta, we also mapped the SLI gene, which we did in an F1 population of 252 plants. We find about 1-to-1 segregation for self-compatibility and self-incompatibility, although many plants could not be determined. And we genotyped this population on chromosomes 2 and 12. And we found a very strong signal for self-compatibility on chromosome 12. We then confirmed this QTL in 2 different populations that share the same self-compatible mother. And luckily, these mother and the paternal grandparents were sequenced, so we could easily design markers. And this time we genotyped only on chromosome 12, and we confirmed that SLI is indeed located at the bottom of chromosome 12. So then we looked at some individual recombinants of this mapping population, and we found a 628 kilobase interval where SLI must be located. Well, this interval contained many genes, and we wanted to narrow it down. So we designed markers flanking this interval and screened 1,374 seedlings with these markers. We obtained 81 recombinants. And by looking at the recombinants themselves, we found 2 which define a new interval of 27 kb containing 5 genes. Although this was already a quite small interval, we repeated the recombinant screening now using markers flanking the smaller interval. At this time, we had to screen, of course, many seedlings, and we found 14 recombinants. And we arrived at an interval of 12.6 kb containing only 2 genes. Well, then we hypothesized that actually SLI is a pollen-expressed gene. So normally when you self a plant that is heterozygous for a trait, you expect 1-to-2-to-1 segregation. But we hypothesized that only pollen with the dominant SLI allele can participate in self-pollination, which would then lead to 1-to-1 segregation for homozygous SLI or heterozygous SLI. So we created an F2 population from a SLI heterozygote and genotyped it with markers alternate in parent 1 and parent 2. And here you can see the results. So in the F2 population, you see no plants at all that are homozygous for the self-incompatible haplotype, meaning that all plants are either heterozygous for SLI or homozygous for SLI. So we confirmed this using RNA sequencing. Here you can see a screenshot of our 12.6 kb mapping interval containing the two genes. And indeed, first of all, we find that only this candidate gene is expressed in pollen. But furthermore, we find that this candidate gene is only expressed in pollen from self-compatible genotypes. So in total, we RNA sequenced 8 self-compatible genotypes and 8 self-incompatible genotypes. And in this figure, we show several pollen-expressed genes on chromosome 12. So most of them show a somewhat mixed pattern between the two groups. However, our candidate gene shows very strong expression in the self-compatible genotypes, but almost completely absent expression in the self-incompatible genotypes. So then we went back to our sequencing data. Here you can see the reads for Solintus mapped to DM4. And we found here a large gap where the Solintus reads wouldn't map anymore to DM4. So we made de novo assemblies, small context for several lines. And what we found was that all self-compatible lines contain a large insertion in the promoter that's not present in the M. So this led us to the model that due to this insertion, this gene can be expressed in pollen, leading to self-compatibility. So this candidate gene is an F-box protein with a PP2 domain. And on BLAST, PP2 domains is described as rather enigmatic. So it's not entirely clear, but in any case, it shows lectin activity, and lectins are hypothesized to be able to recognize glycosylated proteins, and sRNases are highly glycosylated proteins. So to prove that we had really found the gene, we took two approaches. First, we designed a construct containing the SC haplotype of SLE, but without introns, and with its native promoter and terminator. And we use this construct to transform two self-incompatible lines. And on the other side, we designed a CRISPR-Cas construct that targets the first exon of the candidate gene and use that to transform one of our self-compatible lines. So I'm now moving to the results of this. So here we see B666 in the wild type, which is self-incompatible. So you can see the pollen tubes stop growing in the style. But after transformation with the construct, we see full pollen tube growth right up to the end of the style in several independent transgenics. So in B667, another self-incompatible line, actually the self-incompatible reaction is not as strong. So you can see that some of the pollen tubes are able to reach the end. However, it never sets self-berries. And when we transform them with our construct, we can see full pollen tube growth right up to the end. Now going to the other side of the experiment, where we have a self-compatible line, and the wild type shows good pollen tube growth right up to the end of the style. But after knockout of SLE, you see the opposite. So pollens are no longer able to grow completely through the style, and this plant does not set any self-berries. So here I have a quick overview of all this work. So we observed many self-pollinated styles, and indeed we can see the wild type self-compatible line shows high pollen tube growth. Oh, and I should say a serum here means that no pollen tubes were able to reach the end of the style. A 1 means less than 25% were able to reach the end of the style, and a 2 means between 25 and 50% were able to reach the end of the style. A 3 means half to 3/4, and a 4 means 100% of the pollen tubes were able to reach the end of the style. So indeed, our self-compatible wild type shows good pollen tube growth, whereas our self-incompatible wild types show poor pollen tube growth. the SLE knockout shows almost no pollen tube growth, whereas the SLE transgenics show good pollen tube growth towards the end. So in conclusion, we identified the SLI gene using a recombinant screening approach and then confirmed that we had found the right gene through transgenic complementation. We found that the SLI gene is highly expressed in pollen from self-compatible genotypes, and we hypothesized that this is due to an insertion in the promoter. Further, it's an F-box protein, PP2B10. It has an F-box domain and a PP2 domain. And although I did not talk about this much, I would say that SLE is crucial for diploid hybrid potato breeding. So in Solinta, we find in our inbred materials a very strong enrichment for the SLE haplotype on chromosome 12. So this was my presentation. Thank you for listening. Thanks to the organizers for organizing it. I want to quickly point out that Aad van den Burg was essential in this work. He did lots of the bioinformatics analysis. And further, I want to point out that many interns worked on this project. And that was it for me.
Thank you very much, Ernst Jan. And if you have questions, please enter them in the Q&A. Indicate that you have them for Ernst Jan. We're going to move right on to Natalie Kaiser now, and we'll do the questions after Natalie's talk. So, Natalie.
All right. I'm just getting my screen sharing going.
Okay, very good.
All right, and can you see my screen?
Yes.
Super. All right. My name is Natalie Kaiser, and I'm a PhD student in the Potato Genetics and Breeding Program at Michigan State University. I would like to thank the organizers for this opportunity to share today how we're assessing the contribution of SLI to self-compatibility in North American diploid potato germplasm. And I want to highlight first off that this work is part of the Potato 2.0 project that's supported by USDA Specialty Crops funding in collaboration with partners at institutions across the US. And what I really hope that you take away from this talk is that genetic factors contributing to self-compatibility in diploid potato are complex, but markers linked to SLI have utility for several important applications in diploid potato breeding. Solanum chacoense SLI donors, such as inbred clone M6, have been used to integrate self-compatibility and male fertility to diploid germplasm, but introduce as well unadapted traits like stolen production and late maturity and high glycoalkaloid content. So markers that are linked to self-compatibility, SLI-mediated self-compatibility in these Solanum chacoense lines, as well as alternate sources of self-compatibility, would be really beneficial to efficiently develop inbred lines. And recently, as Ernst Jan mentioned, Claude et al. Published 18 competitive allele-specific PCR markers, or Cas markers, that were linked to self-compatibility in 2 diploid Solanum tuberosum mapping populations. And they also showed using whole genome sequencing data that self-compatible SLI haplotypes are present in cultivated tetraploid varieties. So we wanted to use these published markers to answer a couple of questions. Specifically, we wanted to assess the haplotype within the candidate SLI region in self-compatibility donors used in North American diploid breeding programs, as well as in dihaploids that are extracted from cultivated Solanum tuberosum varieties. And we wanted to determine the feasibility of using SLI markers to select for self-compatibility in a diverse diploid recurrent selection population. Now, because these markers were developed and validated in Solanum tuberosum populations, we first selected the 9 markers that were predicted in silico to amplify a single product in M6. And those are shown here in their distribution across the candidate SLI region. And using a diverse set of 335 diploid lines, we found that only 6 of these 9 gave unambiguous calls in over 85% of the tested lines. So going forward with my talk today, I'll be referring to these 6 markers shown in black that were informative. And I just want to highlight that each CASP assay included a self-compatible and a self-incompatible allele-specific forward primer, as well as a common reverse primer. And so when I refer to the self-compatible and self-incompatible genotype, that reflects the sequences that were amplified by these primers. We interrogated the genotype at 6 marker loci in 8 diverse self-compatibility sources. So we used 3 Solanum chacoense lines, M6, 524-8, as well as a self-compatible selection from the PI 133664. And we also examined the genotype of Solanum tuberosum dihaploid USW4 and clone XD3, which has W4 as a parent, clone RH, DMRH89. This is an individual from a cross between DMRH and finally the clone 1S1, which is a Solanum tuberosum group Freya clone from Richard Veilleux at Virginia Tech. And what you're visualizing here are the marker genotypes in order of their physical position, where the self-compatible homozygous genotype is given by AA, and the self-incompatible homozygous genotype is given by BB. And it's important to note that there is a heterozygous haplotype for SLI in Solanum chacoense 524-8, which has been inbred for just as many generations as M6. And I also want to draw your attention to the 3 lines at the bottom of the table here, DMRH 89, 1S1, and this chacoense PI individual that are self-compatible but lack the self-compatible SLI alleles across this region, suggesting that other factors are contributing to self-compatibility in these lines and that they could serve as alternate sources of self-compatibility for breeders looking to expand the genetic base of self-compatibility donors. We then wanted to explore how SLI contributes to self-compatibility in a recurrent selection population that was designed to increase self-compatibility while improving agronomic traits and maintaining genetic diversity. And really briefly, this population was created by former student Maher Asakhalani from 5 self-compatibility donors shown here and a diverse set of individuals from different species. Using a 1-year cycle, individuals were intercrossed in the greenhouse in the winter, planted out in the field in the summer, and then selected in the fall for agronomic traits. And over the course of 5 cycles of recurrent selection, the percentage of self-compatible individuals increased from 13% to 83%. We used 228 individuals from the cycles of this population, and we observed that the self-compatible SLI genotype, which is given by AA here on the y-axis, increased almost to fixation over the course of these 5 cycles of recurrent selection at the 6 marker loci that we tested, which are shown by the colored lines. We then looked at association between marker genotype and self-compatibility under greenhouse conditions, and we found that 5 contiguous markers were positively associated with the self-compatibility phenotype, and those are shown in blue here, while the most distal marker, SLI 424, was not significantly associated with self-compatibility. And of those 5 that are associated with self-compatibility, we observed fairly modest prediction accuracies. So we compared the marker genotype at these 5 loci to the fraction of flowers setting fruit, which is shown on the y-axis here. And we saw that the self-compatible homozygous genotype was only present in self-compatible individuals, but that the heterozygous genotype and the homozygous self-incompatible genotype, which is shown by BB in these graphs, was less reliable. And this can be attributed to the difficulty in phenotyping self-compatibility, as well as genetic features that are diverse, contributing to self-compatibility in this population. We also examined the association of the self-compatible phenotype to SNP marker genotype using SNPs from the potato aluminum array. And we specifically looked at SNPs that were residing in the area of our CAS markers that were associated with self-compatibility in this population, we found 2 SNP markers that were associated with self-compatibility, and these could possibly serve as a selection tool for breeders that are already routinely genotyping clones with the SNP array. And finally, I want to switch gears and talk about the genotypes that we observed in dihaploids extracted from Solanum tuberosum tetraploid varieties. Because nearly all Solanum tuberosum derived dihaploids are male sterile, it's not possible to identify self-compatible clones by self-pollinating them usually. So we used a panel of dihaploids that were extracted from tetraploid varieties and breeding clones and assessed the SLI marker genotype at the 5 loci that were associated with self-compatibility in the recurrent selection population. And what you can see here is that we identified self-compatible alleles in dihaploids extracted from cultivars Superior, breeding line NY148, and cultivar Atlantic. And I also want to highlight that we found a dihaploid that was homozygous for the self-compatible SLI genotype at all marker loci. And this really shows that the SLI haplotype can be used to inform the selection of dihaploids to use in a breeding program to increase the frequency of SLI self-compatibility alleles. So finally, I just want to emphasize that the varied SLI haplotypes in the 8 diploid self-compatible donors we surveyed underscores the genetic complexity of self-compatibility, and the 3 individuals that we found with self-incompatible SLI haplotypes indicate other factors are contributing to self-compatibility in this germplasm. And we also found that the homozygous self-compatible SLI genotype at 5 Cas markers reliably predicted a self-compatible phenotype in a diploid recurrent selection population, and we proposed 2 SNP markers that could also be used for selection of self-compatibility. And we suggest that SLI genotyping can inform dihaploid selection. And with that, I want to thank the individuals in the potato breeding and genetics program that contributed to this work, as well as Dr. Shelley Jansky. And if you're interested in learning more about the many facets of the Potato 2.0 project, you can scan this QR code. It'll take you right to the website of the Potato 2.0 project. And thank you for listening.
Natalie, thank you very much for your presentation. So yes, please do enter your questions. We have time for a couple of questions. First of all, from Stan Ernst Jan, is the insert in the promoter a transposon?
Well, we are still working on that, but it looks like it is a repetitive sequence. Actually, I could show a little bit about this if you want to. I had a few hidden slides that I did not show. Let me see if this works. I have to wait. Share screen. So when we blast the mite or the insertion, we get lots of hits from, first of all, Solintes, but also many other potato cultivars on many chromosomes with high coverage. And when we blast it with the flanking sequences of SLI, then we find that it's quite common throughout potato genomes. So here are the flanks. So this is promoter of SLI, and this is also promoter of SLI. You get many hits for this whole piece of insertion. So there is still some work to be done in determining what exactly it is, but for now, suffice it to say that it is repetitive and it's likely a transposon sequence.
Okay, in the interest of time, I would really encourage everyone to come to the discussion session after the closing. I'd like to right now just move on to our closing session. So I'd like to thank the speakers from this session for their participation. It was a very good session. Actually, I think I might be a little bit ahead of time. I've looked at the schedule. Sorry, everyone. I think we have time for at least one more question. So, Natalie, I have a question for you, and that is, did your Casper marker work on the dihaploids?
Did the Cas markers work in the dihaploids?
Yes.
Yes. So the 5 that I was showing there were robust in dihaploids from the cultivars that we tried.
Ernst Jan, there's another question for you. Great talk from Giovanni. The question is, what is the distribution of sRNAs and SLI genes in different self-compatible and incompatible Solanaceae?
Yeah, that's an excellent question. Unfortunately, I haven't looked very much into that yet.
Okay, and here's one question from Norma Manrique. Ernst, good presentation. My question is, the structure of the Xbox gene considered to be SLI has been compared to the published Xbox proteins associated to self-incompatibility. Is there any pattern of function? And if so, can you identify multiple sRNAs or just identify the sRNA allele in the line where it was identified?
Sorry, I did not get that completely. Is it in the chat?
It is in the chat. It is from Norma Manrique. It is
or in the Q&A?
Yes, in the Q&A. Yes.
Okay. Well, so the Fbox proteins associated with self-incompatibility are S-LOX Fbox proteins. They are quite a lot. They're apart from the Fbox domain, they are completely different than this Fbox protein. And right now, we have pretty good evidence that this Fbox protein that we found can recognize multiple sRNases. The reason is that we don't see extreme in a population which segregates for sRNases. We do not find a huge amount of segregation distortion in the sRNases, which leads us to think that it actually can recognize multiple sRNases. But more study is required to definitively say this.
Okay. A question from Florentin Club. A great talk, Natalie. Did you check more Chacoense accessions with those Cas markers? Do you have an explanation for the presence of a similar haplotype in Chacoense and tuberosum?
So I checked multiple individuals from that PI 133664 with the Cas markers and found that they, for the most part, they all had the self-incompatible haplotype. So this PI, that 133664, segregates for self-compatibility about 1-to-1. And so that answers that. I'm also looking at some other self-compatible Chacoense PIs and screening them with the Cas marker, but I don't have that marker data at this point.
Here, oh, sorry, Natalie, go on.
As far as explanation for similar haplotype in Chacoense and tuberosum, I don't have a good answer. I don't think we know yet the exact gene flow of this haplotype.
Here's a question from Herman Vanek. The SLI gene is expressed in the pollen tube. Is the protein from the sporophyte, the haploid pollen tube, or the gametophyte produced in the microspore mother cell?
Well, I would say that this acts gametophytically because we see this strong segregation distortion in F2 populations. And I would expect if it acts sporophytically that you would not find that. So our explanation for this strong segregation distortion is that only pollen that have the dominant allele in their genome, in their haploid genome, can participate in self-fertilization.
Okay, this one final question, how to overcome some crosses, incompatibility between different species? I guess this is a kind of a general question. I'll send it out to both Ernst, Jan, and Natalie.
Well, that is a difficult thing, especially in potato, I would say. So there are several crossing barriers, one of which is, for instance, EBN number. But it is possible that SLI could help overcome in certain difficult crosses. But I think in generally there are several reproductive barriers in potato which prevent very far crosses.
And there's been some published work on other modifier genes, such as HT, that are more involved in the incompatibility between species.
Yeah.
All right, thank you everyone in this session. I really think it went very well, and I think we are ready now to move on to our closing session. So, Christian.
Okay, thanks, Helen, and thanks everybody. I'm just I won't keep you from your wine and cheese for very long. But I'd just like to make some a very few closing remarks. Can you see my screen?
Yes, we can.
Yes. Okay, great. Yes, as I say, I'm not going to keep you for very long. I hope I've convinced you, and I'm probably preaching to the converted here, that the Solanaceae are really one of the most important, interesting, useful, tastiest, and diverse plant families that are around. So I think I will start by, I hope if I can move my slides, by a word of thanks, especially to the invited speakers, first of all, for their excellent presentations, but also certainly to all of the presenters who submitted and presented their talks, and also to all those ones who did not. And I think I speak for all the organizers of the sessions, how difficult it was to actually make that final choice. We certainly in the development group had quite a struggle and a long discussion about who to choose, because they were all very, very good abstracts. So thanks to everybody who submitted an abstract for presentation. A lot of thanks also to the diverse and many posters that were submitted. They were all really good, and I think that the poster sessions were really interesting and useful to all of us. And last of all, of course, to all the attendees that listened and posed interesting and difficult questions. So we had close to 1,000 participants from nearly 350 institutions right across the world. The map, I think you've seen before as a background to various presenters from all over, all continents apart from Antarctica. So that's brilliant. We got 137 abstracts and 106 posters. And what is more, there were in general around an average of 300 attendees in each session. So I think in that sense, again, my thanks to everybody for participating. Right, now I really want to and need to give great thanks to all the organizers, and that is Glenn, Giovanni, Jose, Sandy, Lucas, Ilan, Jamie, Joyce, Helen, and I'm getting interference here, and Esther for their for organizing the sessions. And special thanks, gold star thanks have to go to Aure and Susie, who were absolutely instrumental in doing the greater part of all the organization and they really worked non-stop in the last couple of weeks to make this all happen technically and administrationally. So great thanks to those two especially. And last not least, to our glorious and eminent inspirer of the whole thing, Danny, who kicked the whole thing off. And so great thanks to Danny, not only for his presentation, but also for getting the ball rolling. And last but not least, to Aaron, who in the background, I think pretty much helped us in lots of technical aspects to make the thing work. Now, let me just very quickly, I'm not going to go into the history, but we've had 16 years of SOL presentation, SOL meetings. They've been going across the whole world. And I think if we look down this list, many of us, at least the old guys, remember the first to the last and have fond memories of all these sessions that crisscrossed across the continents and have been really successful. You know, we strive to have our meetings switching between continents. We still have a continent to go, which is Africa, where we haven't done so well yet. Let me tell you and invite you that this virtual meeting, it was in place of the meeting in Thessaloniki, but that has only been postponed. That will happen next year. We're planning for other meetings, especially in South America, and we're open for other offers in the future. This meeting has been so successful that I think we have to really think about the format of future meetings. And we'd like you to have your say, whether we have annual live meetings as we have done up till now, or annual online meetings, or try and do hybrid meetings, or have an alternation between online and live. And we will send you around a questionnaire to everybody. Auré will send a poll round on lots of aspects of this meeting, but also on the format. And we will be extremely grateful for your views and opinions, both on this meeting and future meeting formats. Now, I'd just like to go very quickly over the Sol Code initiative. This is something that we launched last year to try and get this community or keep this community together, which we of course are. There is a short paper on the SGN site, and that covers informatics sides, genotype, phenotype resources, and material resources that are available to everybody. There are already a number of Sol Code projects, and that is, of course, in the huge progress made in the sequencing of Solanaceae. For example, the potato pan genome is hopefully coming out at the beginning of next year. Many populations are available for distribution analysis by the whole community. Gene expression resources have been and are being developed, such as the tomato gene expression atlas in Cornell. Something similar is being developed for potato tuber development and life cycle. There are many other projects on the go right now that are essentially allied to or part of the Sol Code project. And that includes new phenotyping platforms around the world, such as the one we have in Wageningen and MPEG. But we as Sol chairs, Sol co-chairs are there for you, and we want to facilitate new activities. We will provide letters of support for projects. We will help and advise early stage researchers, and we will facilitate also networking and collaborations. So please get in touch with all of us or any of us to help you in your career and help you in developing new projects in science. Last but not least, I want to make it clear that the SOL 2021 is being organized by Angelos Canelis and Mondia Huizenia and that will be open for people to apply to come. And the website is already open. On that website, you will find further updates on the program, and we all hope that as many people come as possible, and that we will have a great time, and I'm sure we will, in Greece for the next conference. So I will end up by saying on behalf of the co-chairs, the other co-chairs, many thanks for attending this meeting, and many thanks to everybody for making it a great success, and see you in Greece. Okay, and I think with that, I'll hand back to whoever's in the chair right now. Helen, I think we can move on to the discussion section in the different chat rooms.
Yes, please do join us in the discussion group for the briefing session.